who is Dr. Leopold Parts. Leo is a group leader in the Wellcome Sanger Institute, where he works on function of human DNA and interrogation. And today he will be talking about deep learning for computational biology. Thank you.
Hello. Thank you very much for inviting me here. It's my first work trip since COVID, so I'm very excited just to be out of the house. And it's just such a wonderful setting, and I've been very well fed and I had great interactions with many of you, so I feel like there's really a pressure to deliver something that's useful. So I saw from the show behind yesterday that at least half of you have already picked some deep learning models. So I thought, what can I provide that is perhaps more unique and just a bunch of another YouTube video that you can watch. And also know that you're going to have a great tutorial from Intro on Wednesday and Thursday. It's really just the Robin to his background. You'll get lots of practical skills. You can constantly rate on the best teachers and tutors in the university. Is sought after as a consultant and an educator, so you'll have a great time there. So really, I feel like I have a free pass to talk about things that I think are important. So I thought I'll say a little bit about intuition and background to deep learning, just so to bring everybody up to speed also who hasn't applied them in practice. I'll spend most of the time on simply examples from computational biology of how deep learning is used. But half of them our group has worked on to some extent, and another half I've just been forced to think about. There have been about 10 different subfields that are from computational chemistry to images to DNA sequence analysis, where deep learning is used. And I'll try to focus on something that's it's more on the idea level. Like we won't go into performance differences of 0.3% between different small networks, but we'll try to capture the ideas that those networks embody. And then finally, I think I'm the oldest person in the room, which I think it's the first time that's ever happened. So I feel like as the old person in the room, I should say something about sort of battle-earned scars and basically what to do, what not to do. Okay, so why do you use deep learning at all? And there's a promise that now comes, I think, in everybody's minds when people say deep learning, that it's something amazing and awesome. And that promise is based on previously two, and now I give three reasons for this. So first is that deep learning models are very powerful. So there are sort of strong guarantees, mathematical results that make them universal approximators. So if you have a reasonably behaving function, you have some input, some output, and you have some data that from it perhaps, the deep learning network, if you keep adding neurons, ultimately can approximate that function to arbitrary precision. It's a powerful enough class of models that for anything that you want to model, you can do it as well as you might want to. So anyway, it has some infinite power in that sense. So it's a very, very powerful class of models. The second reason why people like it is that it sort of promises a free lunch. Usually you need to spend a lot of time engineering features, thinking over the problem. But deep learning, you shovel the data in, and something magical happens, and great performance comes up. So people like to think that it gets around the problem or solves the problem of feature engineering. And finally, the reason why I actually like it best is that the compute frameworks these days are so flexible and so well engineered that they are actually a basis for a universal computation with data. So both for describing the models and for inferencing. Whereas previously you probably had to go through some pages of maths to, code max likelihood solutions or do some rational inference. Now you can simply define your model in code and execute some more code to get a fit model out and to characterize its properties. So I think that's just a powerful way to think about data science, about modeling, and I think it's the way forward, and that's really why I like deep learning, is that because of these frameworks that allow flexible computation of data. Okay, so so this is the philosophy. I'll say some things now about the content of deep learning, or the framework within which it operates. Much of this is just directly lifted from a review paper we wrote some years ago. It's, I think it's aged reasonably, like the theoretical underpinnings of maths for statistics haven't changed that much over the last 5 years. But the applications will have. So if you feel more information background, there's just additional references, additional thoughts in that review as well. So the context in which deep learning sits is in a canonical machine learning framework where you're given a lot of data, and clinician comes, can you use this for my diagnostics, for example? So the raw data, you first have to go through the tediousness of pre-processing, getting it from the Excel table to compile the limited file, clean out the NAs and text and commas instead of points and all that stuff, to actually have a nice table. Like I'm going to do a matrix. If you do statistics or machine learning, you want a matrix. So you get your raw data into matrix form. Then usually the modeling then takes place not in the raw data space, but in the feature space. So from the clean raw data, you extract something that you think is relevant for a problem, and you get better matrix. For example, that can be motif hits in a sequence. And then you have a model that tries to fit something. In this case, for example, it's a target of having a red or a blue property, and the feature space can have a good separator of those properties. Then we have a fit model, you can evaluate it and have that data and get results. I think many of these things are covered also in different lectures. And then you have the two flavors that are supervised, like you just heard about linear regression and things like that, or unsupervised, like clustering, that you also heard about. And so the bulk of the thinking work usually in machine learning is in the feature extraction. Like the models are quite commonplace. You have a library, sklearn, you just put any machine learning model onto a data set and get the results out. But it's the feature extraction that usually takes the most time and effort. Oh, okay, sorry. It's the data cleaning that usually takes the most time and effort, but it's the feature extraction that takes the modeling time. And so the premise is that in feature space, your targets may be well separated, and your data space they're not. And the deep learning promise is that you simply get shoveled it in, and the feature extraction here is done, so say, semi-automatically. Just because you have this fixed structure of network that automatically picks up what's reasonable to extract from these data. And it's the difference in time spent for this laborious process of, how should they extract the features, how do you encode that one base pair interacts with the other in a loop, but not in a sort of triple helix loop, to just putting data in and getting a sort of hidden variable observation that captures these features. Okay, so one more, so what's actually, there's the machine learning framework, input to output, different ways of doing it. One way of doing it is deep learning, and deep learning is in the end a mathematical function. I like these illustrations that come from a Estonian math book. A function is something that takes an input and produces an output. In this case, it takes a hedgehog and produces a snail, or it can take a distance traveled in a taxi and it gives out the price. But this machine is fixed, and sort of you can think of it as a black box of input and output. And the black box of deep learning has a particular structure. So here is a very simple deep learning network, input layer, one hidden layer, one output layer. The input layer is observed, so it's not hidden. You know what goes in. The consecutive layers are linked to previous layers with weights. And the final output layer is usually, again, a target either self or target value that you want to learn from the input. So the input and output are observed, and everything in between is computed, so it does exist in the computer memory, but in a way, from a modeling perspective, it's a hidden variable. It's a state variable that is not part of the model. And within one neuron, so inspired by cat's brains and other neural experiments in the 50s and 60s, it tries to mimic something that the neuron does, which is to aggregate the total input and either activate or not. And the common structure for these is to simply sum up all the incoming edges. So here, for example, we get every single edge that comes in brings a value, sum up those values, and you put that through an activation function. So in this case, it's a ReLU function, which is constant in the negative and linear in the positive, and that gives them an output. Okay, so it's layers of neurons, link pair connections that have limitable weights, and that ultimately produce an output. And the way learning works here is by trying to best produce the desired outputs. So again, the inputs are fixed, the network structure is fixed. The only things that are variable are these weights and these errors. The way to improve the weights and those errors is to backpropagate the loss. So this implies that everything in the neural network should be differentiable. So this loss function, you have to be able to backpropagate the gradient of the error with respect to the weights. So there has to be ability to push information back. So not all activation functions are really nice choices. And so in a forward pass, it takes some input data, you calculate the output, it's okay, this was reasonable but not great. And in the backward pass, you compute the loss, the difference between expectation and desired value. And and they update the weights according to the gradients. So this is, probably you've seen this 17 times each of you, but then again, so the gradient update, if you have the weight space here and loss here, you take a step in the weight space towards the gradient with some learning rate. So you can tweak how big of a step you take in the weight space. And that's a parameter you have to set in deep learning. But if you take big steps, you can move to great new grounds in the weight space, but you can also sort of lose your current good standing. So it's not obvious what a learning rate to use, and I think the usual guidelines are to use big weights early and then small weights later because you're more likely to be near the local optimum. Okay. And so I thought it's important still to internalize that in the end, neural network, it's a mathematical function, and the form of that function is fixed, and the form of that function determines what your model can capture about the data. And the learning in the network actually brings in additional information because you have to define the loss function. The loss function tells you something about the task. You can have the same network perform multiple tasks by changing the loss or changing the output layer, even if the network structure itself is fixed. And again, I still want to keep reminding that you build these networks by adding neurons, by adding layers, by taking a previously pre-trained network and sort of jamming it in to provide a new capability. You can think of this, this is what programmers do. You add a statement, you add a new function, you're calling this function. So anyway, there's this composition and abstraction that's possible with deep learning. So you can think of it as programming the data. Okay, I think I'm almost done in the philosophy. I just highlight two more examples of operations that are common and are worth internalizing again, and then we'll get on to the examples. The two operations that are most common they use in deep learning and that are worthwhile having a good intuition about are convolution and pooling. So first, convolution, you can think of it as pattern matching. In convolution neural networks, you have neurons that effectively fit a pattern, and it can take a form like this, where the pattern here is the square of 1100. You think of, if you do a dot product of this with other matrices, what does that look like? You're going to have high values if there's high values on the diagonal, and low values if there's not that many values on the diagonal, and perhaps there's something somewhere else. So it's like a diagonal edge detector. And you can assess the fit of this filter or the strength of such signal to a particular place in your input by simply doing the dot product and recording in the next layer. So in this case, if you match this filter to this place in the input, you just multiply it element-wise and add up to get 1 times 1 plus 2 times 1 is 3. Then you can do the convolution operation of keeping the filter fixed, so it's agnostic to the position at which it's applied, and simply sort of slide it across. And so you get something that's slightly lower dimensional because you can only fit it in a 3x3 grid, and then compute the filter match or pattern match at each of these locations. So to the next layer, we pass on how much did I see this type of signal in this location. The other important operation is pooling, like in this case, max pooling. So you can think here of just aggregating signal from an increasingly large region. So here, if you do a 2x2 max pooling from this square here, you just take the maximum value of that square. And you can move over stride 1 or 2, in this case, it's stride 2, then you take this one, okay, the maximum here is 4, maximum is 2, maximum is 4. And the next layer gets information about the strongest pattern match, but in a lower dimensional way than you would have here. So this allows you to play with the dimensionality of data. So here, you can add more neurons, more filters to have a richer representation of the input. And here, you condense that richness down to a small number of variables that you can then use in a sort of more dense way, let's say. And an example here is usually convolutions are best thought of in sequence space or image space. In sequence space, you think of DNA motifs. So you have transcription factor binding site, or transcription factor binding motif, and you see how well does it match our sequences, slide it across. And for image, you can still think in this dot product way. For example, this is your filter, the kind of signal that you're looking for, which is like a diagonal edge detector. You can sort of slide it across here and record the strength of the match at different positions of the image. And so you can see that this brightness towards the bottom left, and darkness towards the top right. That's mostly present, as you would expect, in that area of that image. Then to reduce the dimensionality, then we can do max pooling and get a smaller dimensional representation of this filter field. And usually this combination of fitting filters or fitting features or discovering features, evaluating the strength of the fit and then combining across regions, that's then followed by dense layers. I mean, you can think of this as one layer is like a linear regression or logistic regression, 2 layers is logistic regression with interactors or interaction terms. There's lots of parallels there as well. So it's then just ignoring all the spatial structure and putting it all in, trying to understand, well, trying to regress the desired output. Okay, that's all I want to say about deep learning in general. Just to remind that they're a mathematical function, and the form is fixed, but that fixation of the form, it also fixes what you can actually capture in data. If it's a full dense network, then effectively doing multiple regression of lots of interaction terms. If there's also convolutions, then you're saying that, well, I expect that my data will have patterns that are agnostic to location. If you have long-term memory, then you're saying that I have information that's covariant across long distances. So everything that you put into the definition of the function impacts what you can get out. By the way, this blue dot here is for me to speak slowly. You can see how it's important, but it's not yet doing its job. So, and the network structure itself is fixed, flexible parts of the weights, and that those weights are optimized against the loss function. And that loss function gives you control over the task. It can be quite different from the same data and the same target. How you define the loss, what your model actually learns, and all kinds of errors it makes. And again, I still want to keep back on this drum of general purpose programming as deep learning. It's building a network and choosing the loss. There are ways of programming with data to capture the features that are important in the data, and then also the properties about the task that matter. Okay, so next up are examples. Does anybody want to discuss anything about models in general? Okay, so now about 10 papers worth of deep learning for ... And I have experience with some, but not all of them. But I have hopefully some intuition about most of them. I think AlphaFold. Does anybody know AlphaFold inside out? Yes, you will do that part. I'm excited to kind of learn what it's all about. But the way I want to do these examples is for you guys to also think about what the important characteristics are to that, and so the introverts can think to themselves for some time. The extroverts can talk to their duck or the neighbor to discuss what's a good representation for a particular task. What's the loss function that you would use, and you can also think of what the covariance structure or covariance function between instances could be. So the representation obviously matters for what do we want to capture about this data set? What are the features that matter? How do you think about the problem? Anyway, regardless of whether you use deep learning or not, it's still important to be embedded in the theory of the problem and in the science of the problem. So you should understand, what you should expect. You should understand when your model is doing well, and the way to do that is to just understand the problem features. The loss function defines what performance matters, and it's also important to think that through. So, you get what you train for. And finally, covariance function, that's not an immediate thing that people think about. So what makes two instances similar or dissimilar? But it is sometimes more natural to think in terms of instance similarity or covariance. And if you have that well defined, and there's a duality between sort of covariance and featurization as well. But if you have the covariance well defined, it just opens the door to different types of models. Like for example, you can use this for clustering. It's very natural if you have a distance or covariance measure to use that for spatial clustering. You can use that as an input to kernel methods, which is a sort of powerful class of methods that can be I want to say an adjective that's positive, but not just too disparaging in learning. Kernel methods that are also powerful, and you can just directly optimize it during training as well. You You might want to have similar things to be more similar and dissimilar things to be more dissimilar. And there's just ways for training for that in deep learning. Okay, so my first example is from something that we produce ourselves. I'm not going to let you choose, but you can think of deep learning for classifying protein subcellular localization. So here the inputs are the 64 by 64 pixel image patches that are centered on the yeast cell. They come in two channels, red and green. Red is background, green is fluorescent protein we care about. The target that I want to predict is the localization of that protein. So if you look by eye, here's the nuclear, these blobs. Back here are bigger blobs. Okay, you don't see the nucleolus, which is a sort of crescent within the nucleus. You have peroxisomal dots. You have the ER, which is the nuclear periphery, plus additional membranes, and so on. So, what's a good amount of time? 30 seconds? How much do you like to think? Okay, so think for yourself 40 seconds. Does anybody have 50? No, okay. So 40 seconds to think about, how would you represent these data if you have an image to capture this localization information? What kind of loss function you might use? What kind of covariance function? Go. Three, two, one. Yeah, I take the whiskers as a positive sign. So things that I could think of were here, like, sorry, I shouldn't open this up. So what did you think?
You can use just the, diffuse different colors.
Sorry.
Just an image, like
Yeah.
colors.
3 halves. Like,
Okay, here you have two.
no, but like basically you're right that if you have a cell in the center and you know that nucleus is sort of inside the cell, then you could look for a big blob in the center, for example. And you look for sort of how much signal there is in total. So just overall intensity, number of bright pixels, that will tell you whether it's a small dot or something else.
Maybe something like density.
Density. So how would you encode that? Density of sort of bright pixels in the green channel, or?
Yeah.
But density is sort of a local thing. So would you mean like just an average intensity, or? Like,
for local slicing.
I see. Yeah, so you could ask, other locally dense regions, for example, and that would require defining it. Yeah.
So maybe, for example, okay, I'm not sure how to call this, but the number of given color, the number of distinct clusters, and somehow the shape of those.
Yeah, so that's what the classical solution is going towards, where there's a sort of there's a nice package cell profiler. It's developed by Leopold, and it just extracts lots of image features that's quite agnostic to the problem. It's like a pre-trained feature extractor. And one of the things it can do is try to segment the number of foreground objects in background. So you could say that, well, it seems like the green channel here, if you look at the pixel intensity histogram, there's bright things and dim things. We could just try to fit a bimodal distribution there, take the bright things, then look whether they have one big dot or many small dots or something else. And then that number of objects and their size tells us something about whether it's a peroxisome with lots of small dots or a nucleus with one big dot. So yeah, that kind of information has to enter in a way, and same with density and so on. All of this has to somehow come to the model. But when you try to write it out in maths or even just in code, it's not completely trivial how you do that yourself. So, and then there's more standard signal processing things that you can say, like sort of predefined filters. But yes, right. And this is just stealing from the cell profiler feature list, basically. And loss function. Sorry, I need to learn how to use this. An itchy trigger finger. So, I mean, target is sort of a binary, so on a 1 of k classification. So there are only so many things you could use, but there's subtlety here because, for example, let's see, the spindle pole. So this only happens when a cell is dividing and only a short duration during that. So this is a really rare class. We only have some hundreds of images here, whereas the rest are thousands. So is there a question of whether we care about the loss of accuracy on those images more or less? And you can already encode sort of preference towards a particular class or a preference towards a false positive or a false negative in different classes differently. You can encode this in the loss stage. So one way to deal with the dataset imbalance is to upsample or downsample, so you have uniform coverage. Another is you can just encode it into loss as well. So again, depending on what you put into loss, you could get more false positives for spindle pole, or just because you keep exaggerating their importance, and maybe the model will start liking finding them more. Or you can have more false negatives because, well, you just don't see that many spindle poles, and it's more likely that it's actually protozoa because, well, it's rare to see spindle pole. And the covariance function, I'm not going to stop here because it's not, I couldn't think of a nice covariance function, that's all. So ultimately it will come down to some form of representation. Dotted cells should be similar, but I wouldn't know how to compute the covariance from these cell tiles. So if you do have an idea, come talk to me afterwards, I can, Okay, so the natural model that people use for these types of data, and also use is a random forest or this detective boost if you follow Kaggle, and put this image into cell profiler, just say calculate features, and it'll do, depending on how many modules you use, between 200 and 4,000 features. You put this into anything that's in a scalar and get the predictor out. And we also gave this data to one master student, and that was some years ago already, but basically he fit this convolution neural network, which basically uses the same ideas that you've just seen. 3x3 filters to find patterns, then convolving them also across layers, so filter with filters, so patterns of patterns, then pooling layers that combine the filters across space to four-dimensional things, and so on. So altogether there was 12 layers, so fully connected layers in the end. And final one was with these 12 classes. And just quickly about the performance, on the x-axis are the different classes, on the y-axis recall for all the instances of the class. Red is deep learning hours, all the best. Blue is random forest using self-program feature sets, not as great. And we're quite happy just because the remaining errors, which is make lots of plots, look at what the errors are. We spent so many, I don't want to say years yet, but we definitely spent months trying to optimize models. And when we finally looked at the errors, we just saw that, that's okay, there should be errors. And if anything, we should remove such instances during training. Like all of these, by this performance, all of these were still classified as errors, but really, these should be something like nucleolus or cell periphery, but there's just nothing there really to evaluate. We're also quite okay with having false negatives. So this should be cell periphery, which is just a ring around the cell. And this is not just a ring around the cell. And this also, to me, doesn't look like a ring around the cell. And here at the bottom are false positives. So it shouldn't look like a ring around the cell, but this one to me does, and this one as well. So we're not too unhappy about those as well. And there are just tricky cases. So this is nucleolus. So this is a crescent shape that's around this big, but it's just not completely round. It's more like a half moon. And if you look at the nucleus, which is, nucleolus is part of the nucleus, it's just occluded by ribosomes, not ribosomes, sorry. It's occluded. So it sort of makes sense that nucleus and nucleolus can get mixed up more often. Or if you only see a small punctate, then that's can also be pretty much whatever. So it's okay to have these kinds of errors. And finally, we want to interpret a little bit of what do these individual filters actually see. So here on the y-axis will always be 5 different neurons from the layer that's linked. On the x-axis are 3 inputs that give the maximum activation of that neuron. So 3 by 3 input to these first neurons. Now here you can see, you can interpret these hand waving it. We don't want to say causality. That this is an edge detector, sort of top to bottom, left to right to left, bottom to top, and then just a general intensity calculator. Go to the next layer, and these are now 5 by 5. So if you remember, if you convolve in the 5 by 5 square, you get 1, 2, 3, 1, 2, 3. So it's a 3 by 3 convolution sees a 5 by 5 input. And so this, for 5 neurons, the 3 strongest inducing inputs are, you could argue that this is sort of looking for this corner here, as is this one. This may be looking more about the corner there, maybe there's an edge there, and so on. And as you expand and the receptive field gets larger for each of these neurons, then you start getting edges, round edges, puncta, overlap between green and red intensity, which I don't actually know what that means, and so on. And then once you get to the very end, like by definition almost, if you get to the very end, the strongest activating images should be the class instances. So you just get really nice separation of cell periphery or cytosol or peroxisol, MER, and I don't know, collagen. Let's say the last one. Okay, so basically there are ways to visualize some of that as well, some of the network inputs or network workings. Okay, so really, I just want to have an easy first demonstration of how convolution networks, they can be quite natural. You have local image features, you have sort of puncta and membrane structures and sort of bigger blobs, and you can have filters that detect those and then aggregate the presence of those across distance. And you can do this post-hoc analysis to try to interpret what the individual features mean, or at least what they seem to do in the model. And we had problems with data set imbalance, and there are different ways of dealing with this. So we simply upsampled in the end to have equal representation, but you could also include this in loss. And especially, if you have plenty of data and there's good strong signal, and you start getting accuracies that are close to 99 or 98%, and often the remaining errors are actually, you don't necessarily want your model to be 100% perfect, because the remaining errors are not always errors of the model, they are just impurities of the data. So again, I spent months trying new model architectures, trying more parameters to get performance even better, to lower the error, but then if actually had taken 5 minutes to look at the data, we would have said that, actually we don't need to fix this, it's fine. And okay, so this work's written up in Donald's paper from 2016 already. So okay, so this was about 10 minutes for one example. Good. 9 to go. Okay, what's next? Computational chemistry. So again, 40 seconds. Your inputs are these chemical structures. Your output can be something like a property. Does it inhibit KRAS? Or what's the sort of energy level of the molecule? What's the way to represent this in a computer? What's the way to compute a loss against a property or even just self-encoding? But I guess mostly what I'm interested in is like representation and covariance. Like how do you represent this in a computer, and how do you say that two molecules are similar? I think we talked most of this 40 seconds, so you can have another 40 seconds. And time. Any ideas? Does anybody do computational chemistry here? Does anybody care about molecules? Okay. So you get to say things now.
When I graph, for instance, or the most
Yep.
important groups.
So you know the literature basically. Good. Yes. So you can. Well. Can you say more? Sorry.
What's that?
Can you say more? More words on the same topic.
Well, there are groups that are the most important for the interactions. There's no example.
Okay, like what they have like the lensing ring and what they have like this four pairs. So in a way it's like a fingerprint. You sort of enumerate things that you've previously seen that you know are important. You have like a big vector and then you say, do I see this, do I see this, do I see this, how many times do I see this? Okay. I think that's molecular fingerprinting. So this makes perfect sense, and that's it's been mainstay of these kind of analysis for decades at least. I don't know how long the field exists. And let's see. And the way people have been excited about deep learning in this field is to learn this representation, but not to rely on sort of chemical knowledge and understanding, but also relax that a little bit. And usually there's models that capture both, so basically also fixed features, but also allow flexibility in learning that do a really good job. And so one way to do that is to use graph embeddings. And the idea there is that you have a sort of three-way task. So you still have these hidden states. So this is an embedding. We have a hidden state that describes the actual properties of the molecule, of this atom within that molecule. And that is influenced in three ways. So one is you have the atom itself that influences the hidden state, its hidden state. You have all the bonded neighbors that influence the state of that. Basically, nitrogen in one context is not quite the same as nitrogen in another context, different neighbors. But then you finally have the task representation of Y. So basically, how does the hidden state of this atom in this molecule influence the target properties? And so this suggests, if you think that this is a good way to think about the data, then this suggests two neural networks that first try to, well, anyway, there's an approach that uses two neural networks. One to infer the embedding, so what is the actual, what is the hidden state in a way of every atom within this molecule, and one to learn the link between the molecule's embedding and the target. And the key idea is that you have many of these molecules for training, and the structures change, so you almost never see the same structure multiple times. But what sticks across these molecules is the link between, there is a parametric form that informs hidden state from the atom and from the neighbor. So that parametric form is fixed in structure and it's flexible in parameter values. So deep learning comes into play of learning the influence of the hidden state of one atom on its neighbors and learning the influence of the atom's identity on its state. That's one network that basically embeds the molecule in a graph and infers the properties of that graph. And there's another network that takes the totality of the state of all the atoms in the molecule and uses that to predict the property that you care about. For example, you can just have weights for each of the different hidden state elements and sum them up across all the molecules. And yeah, so, and this is slightly out of order. So, and the covariance function could be something like what makes two molecules similar. They could, on a chemical level, what are the number of shared substructures it has, like a bag of words, bag of substructures model, where you enumerate everything and you see how much they share. This can be just how many atoms they share, like how many carbons, how many nitrogen, and so on. But it can also be benzene rings and so on. And one thing I want to mention, in case anyone's interested, maybe Kaspar probably knows this much better than I do, is a Fisher kernel. So it's, these are really quite different structures, and it's not at all natural to me to think about their similarity. But if you do have a model that has sort of an encoding of these different types of structures, then from the model's perspective, a structure is a structure. It doesn't really matter if they are different to you or or not. So you can evaluate the similarity based on the model. And one way to do that is to look at the gradient at the model fit. So you have your deep learning network, input in, structure in, output out, great. And you have this for one molecule and the other molecule. And your network is right now fixed. So you're not doing any updates, but you can still compute what's the gradient. So you can compute the loss, you can find the gradient of that loss. And in a way, whether or not the two molecules deviate from the loss in a similar way is a very natural way of thinking of, does the model see them in a similar way? One way to think about it is final layer activations, the other way is sort of backpropagation of the loss. So I thought this was an important idea to mention, and 4 sentences are not going to convey the complete intuition, but hopefully, if you thought that this might be something useful for you in the future, you can start reading about it. Ultimately, it comes down to the shape of the energy landscape involved fit and just how a data point fits there. Okay, as you can see, this was a more handshake way part because I've never worked in computational chemistry, but they sort of forced me to think about it a little bit. And the key feature was here that the deep learning is not so obvious. It doesn't parameterize concrete properties of the molecule itself, but rather parameterizes how different molecules share information between atoms and from atoms to the target property. So in a way, like again, there are three sets of weights here to learn. One is sort of weight from the atom to the molecule. There's a weight between different parts of the molecule, and then finally there's a weight to the overall property that you care about. And so it's a really cool field that I feel I would like to do more of. But in the end, every data point, every molecule itself defines a model. So you have each molecule is itself a model in which you can do inference and all these things. But the sharing between molecules, the sharing of those model parameters is what gives power to these models. Anyway, so in general, it's sort of embedding of Latent variables model for structured data. It's called structure to vector. So it's a great paper. It's sort of founding of the field, and it's now been used for molecule property prediction. And I thought in Citro bought like an entire computational chemistry unit in Poland to do this kind of learning for drug discovery. So I'm not sure if any of you work there, but there's careers in it. And in general, you can think of also graph neural networks. How do you propagate properties across things that can be described as networks? Most of the time, I'm a curmudgeon, and I don't like graph neural networks. If you really think that your data are best represented by a graph, then maybe you're not thinking hard enough. But in this instance, maybe there is something to it. So, okay. Next, who's working nanopore data? Some of you made libraries in nanopore here at the school a few years ago, so some of you are lying. I'm not sure which ones. Okay, so here the inputs are current. So how many of you see nanopore instruments are working? Excellent. Yes, so you've seen the future. You work with the cool toys, but so I mean they're magical. You're basically pushing a single molecule through a really tiny pore and reading out currents at really small distances. But ultimately, so this is a DNA sequencing approach where DNA molecule is ratcheted through a pore. So the pore is here. There is 5 bases in the pore at any one time, and there is sampling of the current across the pore that gets recorded. And the current in the pore depends on the bases that are in the pore. So you get current readings, and if the ratchet, like the motor protein here that pushes, I'm not sure if it pushes or pulls. Somebody know? Because it pushed or pulled? Pulled. Okay. So as the motor protein pulls the DNA through, every time a new base enters the pore and the previous base exits, the current changes. So you get these traces of current on the y-axis, time on the x-axis, and the task is to decode this into a base sequence. So what's a natural representation for this, and what's a good loss function? Okay, so for those of you who have dealt with nanopore data, no. Yeah, that makes a lot of sense. That in a way, your state space is finite. You have 4 to the 5 options, and each of them defines some current level. You may not know them, but you can sort of learn them. That's that's basically it. Basically, the finite setup for it. I don't know of anything else really to put there. So yeah, and in loss function is quite direct. You just do I get the right sequence out? And covariance function also can think of anything useful, but there's usually covariance between the currents a long time. So ignore that bit. But dimensional model, and if you have a finite state space and you have transitions in it, the most dimensional model is a hidden Markov model, right? So that's the original base calling model for nanopore was just an HMM. So you have your, back then it was 4 bases in 4. So you have a state like AGGT in a pore, and then the natural transitions are you delete the first base and you add 1 base to the end. So AGGT can become GGTA or GGTT or GGTC or GGTG. You can also just stay in the current state, so the same current, well, current and current, it's overloaded word, it's English language. So you can stay in the state you're currently in. You can stay in the same state, and the current shouldn't change. And sometimes you also just have a stray transition. And so once you have feature HMM, you can then decode it using the standard material algorithm. And that's reasonable, but when people, as recurrent neural nets and others came onto the scene, people started trying those, and they just outperformed. So in a way, you can relax the pure transition matrix representation, where you have, well, in the end, you're just learning transition parameters of a big matrix. So here, should it be evacuating? Okay, no. And frankly, I don't know what the motivation behind this model was, but here one way to relax is to say that every reading is encoded by some state, and you can then convolve across states in consecutive layers. So the first layer only gets 1 reading, second layer gets 3 neighboring readings. So the top one and neighbors. The next layer on gets even more. So by the time you get to the 5th layer, you're expanding sort of almost 10 bases in. So in a way, the capturing covariances that are longer length scales than are actually inside the pore. And then you can add some additional ways to keep memory of what's happened before and after, to clean things up, and then decode. So I guess my takeaway from this was that it's a way to relax the hidden Markov model, where you have it's a very natural representation, but you have a fixed model structure where if the data doesn't quite conform to that model structure, then you're sort of stuck. You have to start modeling exceptions. What happens if the pore stutters or it skips a base, and it can be, especially if you have lots of data, and sequencing machines can generate lots of data, then it can be easier to over-parameterize or have a more complicated model that allows more flexibility in the ways you encode dependency between consecutive data points and to fit that instead. So this models have gone through many iterations. I think the first copy-based caller was in 2018 or so. We've had 5 years to have better and better tricks from machine learning literature to improve the base callers. But knowing that people will talk about NLP tomorrow, I just want to first mention attention already. So this is one way that the current transformative base callers are working for nanopore. And the idea is that instead of having an HMM, where you know exactly what your state transitions are, and therefore what matters from the past, like the Markov properties that the past doesn't matter, instead of having something like a CNN, where you have a longer view of the past, but it's still kind of fixed, you can have almost infinite memory, but you have to pay for that by learning where you need to look. And one way to do that is using attention mechanisms. And roughly, it comes down to learning a matrix that's between encoding and decoding that captures the covariance. How often does decoding this base depend on the encoding of signal in past and future? And that the more length scale you allow there, the more data you need to learn that. And the usual example is taken from Attention is All You Need paper, where this is basically the AIJ matrix for encoding and decoding English sentence. You can see that so the green arrow is basically I is on the bottom and J is at the top. So how much does the decoding of the article the depend on everything else? And basically it only cares about this sort of subject noun. So it's the law, nothing else really matters. Whereas law itself influences the decoding of many things. The law being perfect, and so on. So there's a flexible way to learn which aspects of the input sequence to upweight in decoding. And there's lots of tricks and lots of famous models around this. I'm not even going to try to get into this, but again, it's a useful thing to Google and start reading about it if your data have this sort of sequential form. Okay, so for non-core base calling, on one hand, it's a nicely confined, simple problem of having to find where the core and decoding it. But on the other, it helps to decode even longer range interactions. So this kind of networks, they try to somehow parameterize where you should look on the sequence, like is it close or is it far, and how should you weight those contributions. And at least to me, a useful way of thinking about them is the way of relaxing these more constrained models. But you have to pay a price. So just like if you have a linear model without interaction terms, it's sort of cheap to learn. Once you have all the interaction terms as well, you just need a lot more data to have confident parameter estimates. So also if you need to learn where to put your attention over long time scales or long length scales, you have to have lots more data to be confident that nothing in between matters or something further afield doesn't matter. Yeah, so just an example paper is a review basically of different nanopore callers that go from HMMs to RNNs. I think transformers were a bit later, so it's a good starting point, but there's many more. Okay, so this is 3 out of 10. Okay. DNA variant calling. How many of you work with NGS data in clinical setting and so on? You should care about this. Yeah. What do you use for variant calling?
For structural variants or
For SNPs.
mixed method, right?
Oh, well done. Okay. Okay. Anyway, so the input data here, and correct me if some of this is wrong, at least back in the day, when we're struggling with data sequences, the input data is a set of read mappings to a focal point. So here, for example, it could be, if you care about this particular place here, you have every bit of stretch here is a read that's mapping. The color encodes something about the quality. The dots and commas are mostly just, it's mapping this strand or the other strand. And the stars indicate, insertion or no insertion. And the task is, given this kind of mapping of reads to this location, tell me what actually happened in the sequencing sample. What's the right sequence to put at the top as a reference? Okay, because as I perhaps should have suspected, we don't have that much time. I'm going to get 20 seconds to think, to save time. So people who do variant calling, pick on you. So how would you represent, just this information? I guess you have a file format for it even.
Maybe number of these aligned to some projects.
Yep. Count, and also you usually have the quality metric for them, basically the error probability of them. And you could think about the summaries as well, like if it's hard to map region in general, and so on and so on. Basically, you could put lots of information. And then in loss functions, again, I'm not sure, like when you do variant calling, do you play around with the loss function? Like for clinical grade things, do you overcall variants just to be sure that you don't have any cancer gene mutations, or are you just really conservative not to overcall to not make people worried? Like, how do you strike the balance between precision and recall? Sorry? Okay. Yeah, so it's not true. You can think that just genotyping accuracy, how often do you find the right SNP? That makes a lot of sense. But as soon as you actually need to apply it for a particular purpose, then it turns out that it becomes very important what you do with these corner cases and so on.
What is used as a ground truth in this case?
Sorry?
What is used for training for ground truth?
Yes, what indeed. So, at least back in the day, there was one reference genome, NA12478, I think. Basically, there's one genome that was sequenced over and over and over and over again. So if you were evaluating your base caller or something like that, you would take reference data sets and try to pull SNPs there. And basically people trusted that there's a gold standard set of variants, and then it can evaluate how well you do compared to other base callers there. Variant callers there. But if you have a new sample that you don't know anything about, I mean, I guess ultimately to say something about quality, you need gold standards or replicates. So either you evaluate against the gold standard, or you have multiple experiments of the same thing, and you ask how important they are. But I can't think of anything else off the top of my head. Can anybody?
So this is the long sequence.
Sorry?
This is the long sequence and no sample.
This de novo sequencing assembly quality is a separate lecture
Yeah,
itself.
I know.
So yeah, so this is, but it's a practical problem that just comes up a lot. And I liked it back in the day. Like I don't think publish it ever, but I like it because I like Bayes, and this is just such a great place for Bayes. You have your no mutation model and the mutation model. You have reads that can be generated from the no mutation model, reads that can be generated from the no mutation model, and you sort of have a likelihood function. You multiply the probabilities, and you set a prior in and do posterior inference, and it's great. And I thought, this is optimal. Like obviously you can't do better than Bayes. Like Bayes is probably the best. And then people at Google looked at these pileups instead. It's like, well, when we actually look at evaluate whether we trust a SNP or not, we look at these pictures, and a true SNP would look like something like this. About 50% of the cases, one of the two copies of the gene will have a variant, others won't. A true deletion should look like this, where half of them have a deletion, half of them don't. A true deletion of both alleles should look like that, but then if it's a false SNP, then it should sort of lots of errors everywhere. Maybe that's a bad mapping, and it just looks quite different. And so basically, they just did machine learning directly on images. So their input was an image like this, and their output was, again, is there a variant or not? And so their deep variant were short-circuited all of this, and unfortunately performed better than Bayes. And I'm still not quite over it. I think it's against the natural order of things, but there you go. Like the caller that you use right now, does that use images, or does that actually go back to sequence and do sequence? Okay, good. Okay, good. That phase is over, but it's a creative way of, like, regardless or not whether I like it, it's a creative way of using these data, and ultimately comes down to a network operating in the space that the humans operate in. So the key insight for this was that when the researchers were looking, how do you evaluate whether this SNP is true or not? Basically, they would always bring up these pilot plots, and actually, if that's the signal that matters, maybe we should model the signal directly rather than some proxy for it. And also, if you just think of a single base pair and whether there's high quality basis mapping to it, you don't necessarily think of the context that 30 base pairs away where there's any mutation in the same read. But after you think about it a little bit and after you've seen these images, maybe it's quite easy to convince that actually it matters. If the read has more mutation than you expect, maybe it's not a high quality read, and you figure out more things that to include in the model. So it's, in a way, encoding the problem as a picture allowed solving the problem in the space in which sort of humans were solving it, and never get better. At least that's my hand-waving explanation why it performed better. But I'm glad it gets back to sequencing now. But yeah, that's all nicely written up in some paper as well. Okay, so two big ones coming up: gene regulation and single-cell RNA sequencing. So we're only starting to work on this, so I don't have actually good intuition here, but I'll show one aspect. But so I guess I should be precise. Like there's many models around here that can be fit, so you can predict gene expression, you can predict anything. In this case, we just care about predicting all the chromosome tracks you can see in Ensemble or UCC genome browser. So input is DNA sequence, output is ChIP-seq, DNA-seq, ATAC-seq, HVK4 methylation, and so on and so on. So, what's a good representation? What's a good loss function? What's a good covariance function? Maybe representation, as usual, representation is the most interesting one. Anybody work on regulatory genomics? That's all on favor. Oh. We can't raise your hand to everything. Okay. Any suggestions?
And their presentation, like contribution factor binding side.
Yeah, so I guess those are, you have a database of them, and in principle you could map them, and like that's presence absence, that's something you could compute with the sequence without learning anything. It's a feature, one feature observation. And those two things what?
Sequence composition.
Yeah. Sequence composition. So GC content
Yeah.
and yeah, so it's the lower level than motif composition, but yeah. And that's predictor of many things. So yeah, sequence motifs, protein binding, but also just composition. And I'm not sure, so in a way it's a content state, so the combination of them and other early models, they try to somehow capture whether the entire region is packaged, whether like, some dense or some free, but that's something that already comes from the target as well. So it's not clear whether, when thinking about how the model is data, whether it's actually part of the modeling or whether that should be kept cleanly and separately for the prediction part. Yes, and single track's the only thing I could think of what makes sense for loss function is one is, in the end, it's a regression problem. You're trying to predict these tracks and what's to think about. But you could also think that, whether, and I'm still not sure, like if somebody works in regulatory genomics, I'd like to know whether there's quantitative signal in the peaks or not. So does it matter that there's a peak existing, or does the height of the peak also matter substantially? So can these be binarized or not? And I'm not sure about that. So if somebody has better intuition, then they should share. And yeah, and I mean, what makes sequence similar? In the end, it's similar activity, similar motif composition. And I'll just show one attempt at this. There's lots of activity here, and it's the kind of thing that people from our lab are interviewing at Google for deep learning, so basically cracking this regulatory code. And they have been publishing the most, I think recently, both Google and Calico, these types of models. So this is an early Calico model. This is the Kelly SNG2, I think. But the idea is actually, it's quite simple. So they're trying to capture the motif composition and GC richness and so on. And the only thing that they really struggle with is interactions at a distance. So if you look at these tracks, then the promoter for PLD3 can be here, but there are enhancer elements all the way here or there, which is tens of kilobases away. So there's tens of thousand base pairs. You can't really learn a 10,000 by 10,000 covariance matrix. So you somehow have to capture these interactions at distance. And they use quite standard convolutions. This allows you to learn the motif matches where your factors bind and so on. But the dilated convolutions are a way to address how to look at a distance. And so instead of just having convolutions that one base at a time go further and further, they go further and further exponentially. So they double the distance away at which they look at every layer. So instead of convolving sort of one base at a time, one neighbor at a time, they go 2, 4, 8, 16, and so on. So in 7 layers, they get quite far away from the target. So that's one way to overcome usually the limited range of convolution. And later instantiations of this just use also attention networks and transformers. And again, this is something that I haven't deeply worked in myself. We have a student that tries a lot of things, but that's very local. We don't take long-range interactions into account, so I don't feel like I should be trying to give insights. But the cool thing here is that you don't need many encode projects. You can just use the same genome and take slices of it as training data. But if you talk to use data, basically, the human genome is way too short. Like we need a much bigger genome to properly learn something. And ultimately, I mean, the research is not about just trying all the different models and seeing what sticks and what gets better performance. But the productive research in this field has been understanding what kind of signal has been missing from successful prediction, and then modifying the models to capture that kind of signal. So in a way, it's like a generative process where you generate a model that works reasonably well, and you have some residuals that you can't fit well, so you update the model, you add another block or something like that to get even better at picking. And it comes still through thinking and through work, not just relying on some magic of deep learning. So I mean, modeling with deep learning, it is about trying to find the right structure for the model to get the right representation of the data out. Okay, I hope that by the end of it I will have made this point enough times that you will be sick of it. Okay, so next, single-cell RNA sequencing. So here the inputs are read counts in a cell, so I have many cells and many genes. So I didn't bother plotting everything, so otherwise you can't see the individual dots. So every input vector is anyway all gene expression levels in one cell in count form, and you have many cells and many genes. And the learning targets can be quite different. So I think Gaspar, for example, has worked with timing. So you can try to map these individual cells to a lot of time. You could try to quantify a response to a drug, or you could just try to compress this huge matrix into something more palatable. So you can think 25 seconds, what a good representation could be of sort of single cell gene expression matrix, single cell gene expression vector. Proposals? We think of compression, it's almost coming down to the question of, what's a good representation of cell state? Yeah, basically, at least to me, this is a completely non-trivial representation question, and I spent some years just going around and asking people, if you could measure anything about the cell, what would it be? What would be the vector that tells you the most? And depending on what people worked on, people who worked with metabolism, it's like, I want to have all small molecules. Gene regulatory people, I want to have just the gene expression levels. Protein degradation people, well, I just want to have like protein levels and some ubiquitin ligase concentrations, and so on and so on. So basically, it's completely non-trivial. Like distinguishing state from cell type is also completely non-trivial. So there's no good answer there, but there's many things you could do. If you work with people who actually care about the cell type, usually what they care about cell state is marker gene expression. If you want to know your neurons from your astrocytes, from the glia, from other neural cell types, they care about marker gene expression. So that's one natural state. If you're confident, then you can do things like in human cell atlas type experiments, just assign cell type. I only need one of n label. These are fibroblasts, these are T and natural killer cells. That's my compression, and just that label with it comes a whole gene expression profile. You could just do clustering. You don't need to know already what the cell types are, but you do your favorite clustering method. You can based on t-SNE or UMAP coordinates. You can use known proxies for cell cycle stage. Different genes express at different cell cycle stages, so you can actually infer cell state reasonably well from the expression profile. So there are many ways to compress. And the loss function really depends on what you're after. So if you want good compression, it's like a good autoencoder, maybe a mean squared error. But then if you have count data, then all of a sudden that immediately doesn't apply anymore. So something that's natural in more continuous measurement space, once you get to count data and low counts, the residuals are not normally distributed and so on. So mean squared error doesn't necessarily work. I like the idea of maximum entropy of residuals, for example. That was raised yesterday as well. Like that's one way of doing it, and I'll show an example perhaps that uses that. But basically, there is representation not clear, loss function not clear, and covariance function also not clear. Nothing is clear in single-cell RNA sequencing. Basically, because you can do so many things with it, and it's a complicated type. But here, people do a lot with covariance function. You can think of what makes two cells similar. So you can just ask for correlation or cosine distance of the cells. You can ask for co-clustering. You can do PCN and coordinates. You can ask for same marker genes expressed. Like there's many ways you can think of that there's something about whether two cells are similar or not. And one recent model that we care about because it includes perturbations, so both drug and CRISPR perturbations, is a controlled perturbation autoencoder. And effectively, it tries to do autoencoding, but trying to also capture potential perturbations and additional structure in cell state. And the way it does that, it takes gene expression matrix in and it tries to reproduce it. and it has this sort of bottleneck layer, and you can think of this as PCA. The PCA, you get PCA coordinates, and you can extrapolate again. But in this sort of, let's call it a latent space or of the gene expression matrix, they also encode perturbation state and other cell state. And in a way that they try to, it's a very natural idea that in a way perturbations and everything else, they operate on one hand on all gene expression loads, but on the other, if you have a good representation of cell state, you should just be able to move around in that cell state. And the actual single-cell RNA measurements are just like emissions from that state. They're just projections onto more measurements. But like really, the core of the problem is in the cell state space. And so they try to learn an encoder of perturbations and an encoder of other covariates like cell cycle state that could all function additively in this in the state space. And they use lots of tricks there. Like for example, they try to actually maximize the entropy of the latent space given perturbation state, for example. So basically, they try to make the encoding naive to perturbation state so that almost all the effects of perturbation are captured by that perturbation encoding. And then if you can do that, then you can predict the effects of drugs, combination effects. It's beautiful. It's not clear if it works, but the idea is very natural and it's nice. And it's a lot of engineering to get these types of models to work. So one student would try to fit this. There's a PyTorch model. You can just re-encode it. Work for a few weeks. This is on the x-axis is the time of training. On the y-axis is loss. So loss goes down in time, right? Training and validation. They worked almost as well as you could hope for. And yeah, so he worked nicely. It was a short sort of 1 month rotation project. And so finally on week 3, he also sort of visualized some predictions. And so here's the sort of true gene expression matrix that I visualized. And his predictions were basically, it fit the mean. So it's a beautiful large model of which we'd figure out how to fit the mean in a couple of weeks. This would be where Dragon's like, these are large complicated models and you can fool yourself into thinking that the models are doing a great thing by looking at just the loss, but really you should be making lots and lots of plots of what the model is actually doing. And basically there are bugs that are eventually fixed, but by not looking at the raw data form for the first few weeks, we just lost a lot of time. So in the end we couldn't use it, but just a sort of cautionary tale of if you're fitting these types of models and large complicated models and large data, just keep it grounded in reality as well. Okay. Yes. So one key point I want to make that these are huge data sets and there's lots of literature in deep learning for single-cell sequencing, and it's basically I would urge against getting into that field unless you have a concrete task and result that you cannot do otherwise. There are so many people who've done, I know latent variable models are really cool mathematically, I can fit them, and then you're stuck in evaluating your compression method or your latent variable model against 20 others that are already published. And it's really tricky to prove in advance. Is it efficiency of compression? Is it better at capturing some kind of biological signal? It's usually a post-hoc analysis and it's fairly convincing and can be quite frustrating to prove superiority. So instead of just, we could fit this and maybe it'd be cool to fit this, you should strive to learn practical problems, strive to solve practical problems. We don't need one more PCA. Yes, but I'm saying this because we have lots of students who come into our group, I'd like to, build some deep learning model. If it doesn't work, and it's great, they learn how to engineer and they can sort of take that model forward. If it does work, they get really attached and they want to sort of show that it's the best in the world, and which is understandable, but it's a painful path. Anyway, so yeah, I don't want to dissuade you. You're young and full of energy, but it's just full of dragons, that part of the world. Okay, so I thought I'd leave 15 minutes for sort of final words of the old man and questions and answers. So basically, this means maybe one more data set, or data type. So I can offer, let's see, protein folding that I know nothing about. Okay, so can you just quickly then take us through, what's a good...
What's what?
Protein folding, there are people who are very keen and who know AlphaFold. I don't. So I have basic intuition, but I don't. So it would be great to learn.
No, I also don't really know it. And I've
seen people using it.
Yes. Okay. So maybe we can. I want to use one more then that's actually people understand well. But then ultimately, the representation that's usually used is some kind of multiple sequence alignment, and I think the newer iterations of this can also do without, or maybe not this, but they're similar models. And I mean, representation is.
Can I ask for evolutionary coupling, please?
Sorry?
What evolutionary coupling, please?
Evolutionary coupling is the non-independence of amino acid residues at different positions in the protein. So the idea is that if these are the same protein in many different species, if you see, for example, let's see if is there anything here that looks like they're coupled? I don't know. Like maybe this purple and this purple. Like if you have purple here, you don't have purple here. Basically, it's the non-independence of values at different positions. So that's a coupling. And if you see lack of independence in two columns here, that suggests that, well, there's something that's constraining their independent evolution, and that constraint is reasonably hypothesized to be like a physical interaction. So if you see things that co-evolve, then in protein folding, that's been used as evidence for being nearby each other. So in a way, like if you think of doing an all against all covariance here, so you calculate correlation of this column against this column, and this column against this column, places where you see correlation or covariance, they should also be places that are nearby in 3D space. So that and the more species you add, the more evidence you get for this. So that idea, I think Denny Lawrence was the first to use it in 2008 or so. Yes, so that's like you can use evolution information, and it feels like a bit cheating. You know, you're not just using pure biophysics. It's not really solving the protein folding problem, but just carry lots of information. Okay, so for those of you who understand AlphaFold well, as much as I understand, there is two types of signal that the network operates on. One is the multiple sequence alignment, and my intuition around that was that it just does a better job of cleaning up the multiple sequence alignment. Like there could be errors here. It's not clear, you know, where the left and right hand side should be. You may be equally confident by looking at this text on the importance of each of these columns, and maybe a statistical model that's sufficiently powered can give more weight and attention to some of them. So my intuition is that part of it is cleaning up the multiple sequence alignment, and there's another one that's basically what residues can and should be interacting. But now over to the sorry. So does anybody else have more intuition of what actually goes on behind the hood, or shall we do that? Kasha left. Oh, clever. You said yes, you know AlphaFold. You were sitting here and said you know AlphaFold. No, sorry. Then sorry. Okay. If anybody knows, please teach me in the coffee break or lunchtime. I'd like to understand what's going on there. So I have, I think, three more. One is conic self-fluidity, so image analysis. We have histopathology image analysis, which is about the utility of downloading large networks and applying them, and an antibiotic resistance prediction, which is basically if you just apply a dense neural network, it doesn't really do much better than XGBoost. But maybe I already covered all the key elements there. So anyway, so I'm happy to talk to you individually in groups about these other types of data as well. The key insights I want to deliver from CellCon in images is that, you know, if you want to find nuclei here, it's a lot better to do. It's easy to do a lot better than humans because humans can't find nuclei here, but there's signal nodes that can help do that. And we tested many networks for doing that, and they all pretty much perform the same. Like ultimately, if you have a good enough network to capture a signal, it doesn't really matter. But it's cool that you can take individual blocks like these exception blocks or dense blocks and sort of plug and play into different backbones. So there is this modularity again, which comes with like this abstraction and composition power of modern deep learning frameworks. That it's quite trivial to take particular sub-modules of deep learning networks and sort of plug and play them. They may not make much of a difference, but at least it's doable. And also just, you know, if you go from 1 million parameters to 41 million parameters, you increase the carbon footprint, but you don't really improve the performance necessarily. So, yeah, so I just want to say that it's reasonably easy to reuse ideas from from new cool papers, but if you're already doing quite well, it's hard to improve substantially by having much more difficult or much more complicated structures. For histopathology image analysis, I just want to say that being lazy is completely nice. So generalize these types of images, like quite many of them, and there are gene expression data for them as well. What do you do? Like what scale do you do that? Eventually, we just basically download an exception at 3, use that to get features out, use PCA to get gene expression features out and correlate them, and it worked beautifully. So you can sort of save yourself a lot of trouble by not building networks from scratch, but just using huge pre-trained networks as feature extractors. And also maybe even fine-tuning a little bit, and they work really nicely. And for predicting antibiotic resistance, you have the whole genome of bacterium. Is it resistant to antibiotic or not? So we did tests of different types of models, from gradient boosted decision trees to deep learning. So every bar here is performance of a model in different colors, different bars, x-axis different antibiotics, y-axis prediction accuracy, and deep learning models were universally the worst. So basically this is plugging a dense deep learning model onto a set of features that we have manually curated from the genome. So basically this was like gene content, genetic variants, DNA chimeras, and things like that. And just the fully connected deep learning model didn't do very well, or compared to competition. So it's it feels like 10 years ago when people were doing machine learning for a new problem, they would take maybe random forests, maybe SVMs, and use that. And these days it's I just fire up some densely connected neural network and see what it does. And if you look at Kaggle, XGBoost I think still rules in terms of overall performance. So if you just need to solve a simple problem, just use something that's simple as well. You don't need to necessarily build a full deep learning network for that. Okay, I thought there were lots of time left over, so we can discuss your research as well, but no, this is all about my presentation. So, but seriously, if you have ideas and thoughts, I'm very excited to talk about science in general, so I'm happy to discuss these over lunches and dinners. Okay, so I guess on the meta level, I want to just convey that deep learning is naturally used in all areas of computational biology because of its promise for power and just presence of lots of data everywhere. And there's lots of cool ideas of attention, convolution. It's still a cool idea, and others that just allow to better capture signal from the data that sort of match naturally what the data generating process is. But it's important to just appreciate that whether you like it or not, if you define a network structure and define a loss, then you're defining what it's able to capture from the data and what it's optimizing for. So you should be aware of that. And there's this somewhat exchangeability between what to think about. So covariance function and what makes samples similar, there's duality to feature representation. And every covariance matrix is also a dot product of two feature vectors. And so you can think of them as two sides of the same coin, and just sometimes it's more natural to work in one space, sometimes in the other. But people, I think about covariance less frequently, but you should. Okay, so 5.5 minutes by my clock. So of course, Kevin, I'm the oldest person in the room, and I think I read this yesterday, and it just really struck a chord. It's from Douglas Adams that everything that was present when you were born is it's natural, and that's how things should be. I know that Bayes' theorem and just using posterior inference, null hypothesis significant testing, generalized linear models, PCA, decision trees, these are just the natural fabric of modeling in any walk of life, and they should be used, and they're good, and they're sort of like grandmother stew. It's a good thing. And there's the really exciting things that we went between year 15 and 35. These are support vector machines, are great. Random forests, Gaussian processes, deep learning. You should get into random forests while you can. But then anything that's after 35, that's just it's not natural. Like AlphaFold, it's not quite normal. So I don't trust it. I don't understand how it works. So yeah. So I think I've sort of finished this curve, and so basically take all the next bullet points with these caveats that you can do maybe better. Okay, when to use deep learning, it's very easy to use deep learning if you care about performance, but not necessarily understanding it. And so you didn't see the cell counting example in depth, but for example, in one of the collaborations with PerkinElmer, they just want to count cells and images, like that's their phenotyping from the microscope. And we have metrics, we can do performance. We don't really care what it finds in those bright field images, whether it's texture or if it's a retraction of the distance. We have something that works well, and we can do it. You could also think of using deep learning when it's clear that you have lots of data and it's in very high dimensional space, like single cell gene expression data. And if you don't have one of the two, then if you don't have many dimensions, then it's just an easy problem. It's just clear statistics. If you don't have large data, then you should not try to fit very large models. So that's quite clear. The thing that I've been going on and on is exploration of general purpose computation. So you can also search for automated statistician, which is sort of grammar around kernels, and it automatically writes reports about data, which is great. But like an example was in a recent paper from Ben Lehner's lab. So this was just using deep intergenesis protein folding, and they write out this beautiful biochemistry. You have protein that's a ligand binding. First, the protein has to fold, and then it has to bind, and there's constants that sort of govern the balances. And there is some biochemistry that's worked out centuries ago that describes this together with probabilities of something that's bound and so on. And the mutations to the protein, they influence these balances. And so rather to try to fit for every mutation in the protein a different model, and you do the maths and do the posterior inference, they encoded basically a fairly simple model in keras, and then just said infer, and they got the mutation effects of every variant that they tested. So in a way it's becoming mainstream to also just encode general inference, not even deep learning in these frameworks, and it's a flexible, natural way of doing so. So again, I'm not sure if it's deep learning, but you can try to use this tool in this way. And I like that Google is doing all the carbon footprint and training, InceptionNet, AlphaFold, Informer, Localizer. So I say, you can all download them, you can all apply them. I don't know about Informer. But that's where the problem is also that some of them are becoming proprietary. Google has spent a lot of time for AlphaFold, so they release maybe the predictions of the entire proteome, but they don't necessarily release the model so you can predict, for example, which molecules will bind particular proteins so you can find a drug against them. So there's both good and bad side, but definitely it's overall net positive that there are more profit models that are publicly available that you can play with. And you shouldn't perhaps it's a reasonable starting point to download something that's other people have trained and either use it directly as a feature extractor or just fine-tune slightly to see how it works. Probably saves you a lot of time. And so places where we've seen dragons, which means sort of lots of time wasted. We have some data, but not a lot of parameters. Like we have some CRISPR screens with 20 base pairs, 10,000 data points. People have tried to use deep learning there against my recommendation, and just nothing really happens. Like you can save time by just focusing on the problem. People waste a lot of time when they're playing with the deep learning hammer without really understanding the nails well. So what you actually want out of the model, it's important to appreciate that. You have to be able to examine whether the model is doing well, so lots of plots rooted in real data. And at some point you just have to start worrying about the outliers because they give a lot of signal in that application. If you're reading papers, it's easy to get excited and AlphaFold can solve everything. And if you actually try to implement them, you realize that maybe this parameter setting was super tweaked to this data set, and as soon as you go even slightly away from there, it falls back to the baseline level. So treat publications as a reasonable starting point to start exploring from, but don't expect greatness right away. Just to try this point at home, just make lots of plots and make them root in real data to understand well, otherwise you will suffer. And I think in general, advocate thinking deeply about representation. So, in the end, usually if you're doing science, you're trying to understand, and understanding representation is then very important. Okay, this is the last slide. Thank you very much. Do deep learning or do it as a general purpose computation. Oh, can this duck? I like the duck. It failed completely to slow me down, but a nice way to save time is to convince your duck that you know what you're doing. That's that you're making plots, that you're trying a few random seeds if your model is doing well. You add some noise to input to make sure that it's not data set overfitting, and just try a few parameter settings that your analysis and modeling is robust to choices. Questions?
I would like to give it to you
Thank you.
as a thank you.
Is it
DNA.
correctly handed?
What?
Is it right-handed?
Oh, I don't know.
Before I can afford to take pictures of this, like I need to double check the
Yeah.
localizing if you think.
I wanted to do a tattoo with DNA, but I never know which one looks correctly, and I didn't want someone to
Makes
point
sense,
out at the
yeah.
conference, oh, actually, you yourself wrong. But thank you very much for that.